Hello, this recording is for Biology 1406, Chapter 3, Biological Macromolecules. So you should know that there are four major classes of macromolecules, and a macromolecule is simply large organic molecules that make up the bulk of the organic matter in living organisms. We classify them in four major groups based on their chemical composition and shared chemical characteristics. So the first one is carbohydrates. You've heard the term carbs. That's what this is. Sugars is another name for a lot of these. Usually the things that taste sweet are carbohydrates. But the class is a larger group that includes things that don't taste sweet to our tongue but have the same chemicals characteristics. As the name implies, they are a combination of carbon and water, thus carbohydrate, CHO. Okay. Then we have lipids, which are fats and oils, and a number of molecules that function as hormones, and a key component of cell membranes, cholesterol. All of those are in the group lipids, and they consist primarily of carbon and hydrogen. So hydrocarbons are the inorganic, or not inorganic, but the purified form of lipids that we're familiar with from fossil fuels. So all lipids share the characteristic of being comprised primarily of carbon and hydrogen, and then a certain amount of other elements in key locations. Proteins, y'all are all familiar with proteins as being, a major structural component of living organisms, and also a important source of energy and raw material for assembling the other macromolecules. Amino acids are chained together to make proteins. And then finally nucleic acids, which are DNA, RNA, ATP, AGP, and a few other AGP, GTP, guanosine triphosphate, and a few other energy transfer molecules. So DNA, RNA, ATP, GTP, as we'll see in the metabolism chapter, FADH, and other energy-moving molecules. All right, those are the nucleic acids. So all of these are organic molecules that contain carbon. All right, they're also going to have hydrogen, and then depending on the specifics, oxygen, nitrogen, and sulfur and phosphorus. We'll see in these other, used in different ways in these four different organic macromolecules. All right. So the key thing about macromolecules, about all four of these things, is that they're composed of monomers, single units. Think of Legos, like in Lego building blocks, that can be chained together to make larger and larger structures. So in this case, we've got two sugar molecules right here, 6 carbon sugars that have been joined together to make a disaccharide. Di meaning two, and saccharide being a generic term for a sugar. All right. And so all of the macromolecules consist of monomers that can be joined together to make dimers or trimers or quaternary, ultimately polymers, which just means any number of monomers chained together. So poly just refers to the larger chains. You don't need to worry about whether you've said dimers or trimers. Just say polymers for all of them. Specific cases are disaccharides. That is a dimer. It consists of two single pieces put together. And in all cases, the chemistry that joins them together is called dehydration synthesis, because as is indicated right here, we take out the atomic components of the molecule water from two different monomers to join them together. And, we literally produce a water molecule on the side. So we actually produce water molecules as we form these chains by stripping off a hydrogen off of this hydroxyl group right here, taking this hydroxyl group off completely, and then this carbon is bound to that oxygen. There is the illustration right there. In this case, the drawing's a little bad. They did this OH group right here over in this drawing, but here they highlighted this OH right here. It doesn't matter. The chemistry is still generally right. Right? And the bond between the two monomers, you can see these are straight lines, is a covalent bond, so a relatively strong chemical bond. The reverse also happens. We have what's called dehydration synthesis, where hydrolysis. We have the hydrolysis, which is the process of breaking them breaking these bonds apart. And in that case, we take a water molecule and insert it. We break open the joint right here between a carbon and an oxygen, and then we satisfy the valence chemistry of the carbon, and we satisfy the valence chemistry of the oxygen by adding the components of a water molecule to each of those in the form of a hydroxyl group in one place and a hydrogen atom in another place to form two hydroxyl groups right here. And that is hydrolysis, splitting. Lysis means to split, and hydro means using water to split the two monomers. All right, so dehydration synthesis and hydrolysis are the two opposites of that process. And this should not say dehydration reaction. This should be a hydration reaction. So there's a couple of errors in our textbook that came on over with the PowerPoint. This is a hydration reaction. All right, here's an animation that just shows that process at work. Let's see if we can make it play. It's not going to do it right away, so we won't worry about it. Chemical reactions involving macromolecules are catalyzed by enzymes. So this is generally true for all the big chemical reactions taking place inside your cells. There's going to be some enzyme, which is almost inevitably a protein, sometimes RNA, but almost always it's going to be a protein. So for your purposes, just say enzymes are proteins that catalyze those chemical reactions. So catalyzing is a term from chemistry. Typically a metal is like a sheet of metal, ends up being a really excellent place for a particular chemical reaction to occur on that surface. The metal atoms make the chemical reaction more likely. It happens more quickly. That's what happens with a catalytic converter in your exhaust system of your car. And biological molecules, enzymes, catalyze chemical reactions. It's just there's a great big protein typically holding a metal atom in a key location, and the chemistry occurs wherever that metal atom is. And so every one of these reactions, including the dehydration synthesis reactions and the hydrolytic breakdown reactions, are going to be catalyzed by an enzyme. In your digestive system, your digestive enzymes do the hydrolytic reactions of taking complex molecules and breaking them down into monomers via hydrolysis or hydration reactions. Remember, forget the D right here. That was an accident. All right. And so, yeah, there's a heck of a lot of enzymes right here. Generally one for each class of chemical reaction. So there's an awful lot of enzymes in our biology. Okay. And their names, they're usually named after the substrate that they act on, and then they have the word ACE at the end. So amylase is an enzyme, that's the ASE, that acts on amylopectin, which is a particular form of starch. Sucrase is an enzyme that breaks sucrose apart. Lactase is an enzyme that breaks lactose apart, maltase, etc., etc., etc. Same thing for lipases, same thing for pepsidases. These are all enzymes that break down their specific substrate. The substrate is just a fancy word for the chemical that an enzyme acts on. All right. Oh, one other very important idea. Generally speaking, dehydration synthesis reactions, where we take two monomers and put them together to make a bigger, more complex molecule, those usually require energy. So they take energy. ATP inevitably, almost inevitably, will be the thing that pays for that reaction. On the other hand, hydrolytic reactions oftentimes release energy. So much so sometimes that we can recharge ATP molecules, recreate them after they've been spent by pairing them with a hydrolytic reaction. But the key thing is hydrolytic reactions, taking a big molecule and breaking it apart, releases energy. Breaking bonds always releases energy. So let's talk about carbohydrates. Y'all all know that carbohydrates are your primary source of energy for metabolism. And that's what we primarily use them for. All right. If we look at the diversity of carbohydrates, there's three major groups that we find in all living things. Monosaccharides, so that's the individual Lego block. Glucose is a monosaccharide. Fructose is a monosaccharide. Ribose is a monosaccharide. Any of those words with an ose at the end, that's some sort of simple sugar. All right, and the monosaccharides are the ones that we typically use, directly in metabolism. We generate a pile of ATP using monosaccharides. We also use monosaccharides to build more complex molecules like these disaccharides and these polysaccharides. All right. A thing about the monosaccharides is that they always have this chemical ratio of 1 to 2 to 1 of carbon, hydrogen, and oxygen. So for instance, glucose is C6, and since there's this formula of 1 to 2 to 1, that allows you to predict the rest of the formula. If there's 6 carbons in glucose, there must be 12 hydrogens, and there must be 6 oxygens, and that's the formula for glucose, C6H12O6. All right. So the monosaccharides, the ones that are used in biological systems, range from 3 to 7 carbons, with 6 carbons, 5 and 3 being the most used carbohydrates, monosaccharide carbohydrates. All right. Inevitably, they've got a C double bonded to an oxygen somewhere in them. That is the carbonyl group, and then they have hydroxyl groups, OHs, all over the place. Those are the key characteristics of all carbohydrates. All right. And then we've got some names right here. So 3-carbon sugars are called trioses, 5-carbon sugars are called pentoses, and 6-carbon sugars are called hexoses. So that's just a name for the class of sugars, right? So glucose, which has 6 carbons in it, is a specific example of a hexose. Ribose, which has 5 carbons in it, is a particular example of a pentose. And then also, on the sugars, the position of the carbonyl group, the CO, C double bonded to an oxygen, the position determines whether you've got an aldose or a ketose or an aldose sugar. So an aldose, the double bonded oxygen is at the end of the chain. In a ketose, it's somewhere in the middle of the chain. So here's some examples. Here's a aldose. The double bonded oxygen is at the end of this 3-carbon chain. This is a 3-carbon sugar. Notice the hydroxyl groups and the carbonyl group. Here's a ketose sugar, ose for sugar, like glucose, fructose, ketose, aldose, all with an ose at the end, and this is in the middle of the chain, so that's what makes it a ketose. And there's some metabolic importance to the difference between these. Here's a glyceraldehyde, all right, that's a triose sugar. Glyceraldehyde's an important structural sugar in you, an intermediary in a bunch of metabolic steps. Here's ribose, the 5-carbon sugar, and here's glucose, the 6-carbon sugar. All right, so here's a case where we've got 3 different hexose sugars, glucose, galactose, and fructose. They all have the same formula, C6H12O6, but the structural formulas are different, right? So that's why they're isomers of each other, and it depends on where the hydroxyl groups are relative to these carbons, so they can be spun around in different ways. These two are very easy to switch back and forth. This one is much more fixed in its different geometry because the double bond right here is in a different location than it is in these twos. When you've got glucose and you have it dried, you automatically have a certain amount of galactose in there because it's really easy for this to switch over to this and this to switch over. But fructose is a chemically distinct sugar because of the position of the double bond here. All right. When you put those 6 and 5 carbon sugars in water, the molecules are long enough that they actually fold back on themselves and form this ring structure. So in water, 5 and 6 carbon and 7, and if there is such a thing, 8 carbon, the bigger, longer chains, all will loop back on themselves and form this loop structure right here. A 3-carbon sugar is too short. It can't bend enough to do this. So here's some 5-carbon rings, and 6-carbon rings, and here's some 5-carbon rings, ribose, that you would see in ribonucleic acid in RNA, or deoxyribose that you would see in DNA. But that's not this. This is fructose right here, with the 6th carbon over here, an alternative version of fructose. All right. So again, how do you make a disaccharide? You take two single sugars and join them together by pulling out the components. Now they're placed in the correct locations of a water molecule. You make this bond called a glycosidic bond. Glycosidic for adding two glucoses together. And that's the name for this bond in all sugars. It's always referred to as a glycosidic bond. It is the particular dehydration synthesis reaction that gets you a disaccharide. All right? And chemists, have specific names for the location of that bond. We're not going to worry about that. I'm not going to test you on this, but it is important if you're interested in the structure of these molecules and how they fit together. All right? So I told you that the monosaccharides are primarily used as raw material for generating ATP. That's the fuel for your body. And also the monosaccharides are used as building blocks to make other more complex molecules. So a couple of examples of the more complex molecules we make are the disaccharides. There are three that you need to know. Maltose, which is you find primarily in seeds of plants. That's why it's called grain sugar. This is primarily found in plant seeds. Lactose, which comes in the milk of mammals, so not widely distributed out there, and not found in plants, not found in other animals, but it is found in mammals. And then sucrose, the disaccharide that's primarily transported around in the vascular tissue of plants, so it's the most abundant disaccharide in plants. All three of these are used as a way of storing and moving sugar. Storing sugar in a seed until it's needed when the seed germinates, storing sugar in breasts and getting that sugar undigested over to a baby mammal via milk, and transporting sugar around in a plant in a form that the cells along the way can't immediately use. That's the purpose of sucrose. So the disaccharides are usually a way of short-term storage of sugar and transport of sugar. And here's an example of that molecule, of that molecule, and of that molecule right there. All right. The polysaccharides simply are more than 2 or 3 or 4, and typically hundreds, even thousands, of monomers long, the polysaccharides of sugars. These will not taste sweet on your tongue because they don't fit into the taste receptors at a chemical level and can't trigger them. So even though they are technically made out of glucose often, we can't taste them as being sweet. All right? And they're very large, and the purpose of these polysaccharides is either longer-term energy storage or structural, and we'll see that there's different possibilities. Here in this particular case, we've got glycogen, which is something found in almost all animals. All animals store glycogen somewhere, and it is a polymer of glucose that is a long-term way of storing sugar in cells for those times when the cells can't get sugars from anywhere else around them. Then they can dip into their reserves of glycogen, break them down, release the glucose, and use that as an energy source. All right? Something you're very familiar with from a lot of plants is starch. Starch is a polymer of glucose that's used as a longer-term way of storing sugar in plants. Think of a potato. It is primarily made out of starch. The storage roots of most plants. And all plants store some amount of starch somewhere in some cells in them, even if it's not a great big structure like a potato. There is starch scattered around in every plant to various degrees. The ones that we eat are specialists in storing large amounts of starch, and we've actually raised them to be extra good at that process. There are two major forms of starch, a monofilamentous form called amylopectin, amylose and amylopectin, called amylose, and then amylopectin, which is a branched form of starch, where we see that one chain of glucose monomers can be attached to another chain of glucose monomers. That's the branches right here. And these things get to be really, really big. They don't dissolve in water very well. Very, very large, complex molecules. Energy storage in plants, just like glycogen, is energy storage in animals. Same idea, slightly different formulation. Glucose polymer called glycogen, slightly different glucose polymer called starch. Amylose or amylopectin, depending on whether it's single or branched. Here's some other examples. You're familiar with this one. Take glucose and pair it together in an alternating pattern, and instead of starch, you now have cellulose, which is incredibly strong, structurally very, very strong, and very hard to digest. Very few organisms produce an enzyme that can break cellulose, that can break these joints apart because of the alternating nature. They don't fit into the active pockets in the enzymes that break apart starch or the enzymes that break apart glycogen. These things are made to be easy to break apart. So there's a lot of enzymes that get right at them and break them apart to release the glucose. Cellulose, if you want, was made by plants to not be easy to digest. Even though it's made out of nothing but glucose, it's just pure sugar, the cellulose is indigestible by most animals, including us. We cannot digest cellulose. This is the fiber in your diet. It's important, but not as food. Another polymer of glucose that includes some amino acids on it, so it's a slightly more complex polymer of sugar, is chitin, and that forms the exoskeleton of arthropods. This bee, for instance, right here, a shrimp, same thing. And it also forms the exoskeleton of fungal cells. All fungi have a cell wall, not exoskeleton, I'm sorry, a cell wall made out of chitin. Animals make a many cells make a continuous sheet of chitin, and that's the exoskeleton around, this bee, for example. It's not the cell walls of this animal surrounded. Animals don't have cell walls, but they have a collection of cells that can secrete a sheet of stuff, and that stuff glues together, and we call it chitin. Their exoskeleton. So, a little bit about carbohydrates. Remember, you've got to be able to talk about the fact that there are 3 different major groups of carbohydrates that are biologically important. Monosaccharides, ready energy and raw material to build other things. Disaccharides, usually a means of storing and transporting sugar from one place to another. Polysaccharides, usually a means of storing sugar for a longer period of time, or, in the case of cellulose and chitin, a means of creating protective structures. All right? Don't forget the generic formula for carbohydrates, 1 to 2 to 1 of carbon, hydrogen, oxygen. Okay. Then we've got particular ones. In the world of biology, the 3 major classes of monosaccharides are going to be trioses, pentoses, and hexoses, 3, 5, and 6 carbon sugars. All right. The presence of a double bond is true for all sugars. The location of that double bond is what distinguishes aldoses from ketoses. That's this image right here. All right. Here's a triose, a pentose, and a hexose. We can have isomers. Same formula, different structure. And then we get into details. Remember, the 5 and 6 carbon sugars are linear when they're dry, like powdered, crystallized sugar in your kitchen cabinet. As glucose and fructose joined together to make sucrose, but it's in these, straight lines here. But when you dissolve it in water, they always make these rings. And in the case of sucrose, it's going to be the glucose and fructose joined together as 2 rings right here, this disaccharide. Glucose and fructose joined together. All right. Lipids. So remember, the lipids are primarily hydrocarbons, which just literally means hydrogen and carbon, right? So their formula is primarily HC, or CH, I'm sorry. CH, right? And there'll be more hydrogens always than there will be carbons, because remember, each carbon can attach to 4 other things, including so a central carbon in a hydrocarbon is going to be attached to 2 other carbons and 2 hydrogens. The carbons at the end can be attached to up to 3 hydrogens. So there'll always be a few more hydrogens than carbons in a hydrocarbon. All right. Hydrogen, those are nonpolar joints, covalent bonds. So lipids are typically not very soluble in water. Oil is used as a waterproofing substance on the fur of this otter. That's the whole point of the picture. All right, hydrophobic, water-fearing. It doesn't really mean they fear water, but they don't chemically react with water. They tend to be excluded from water. Water tends to be excluded from them. What do we do with lipids? Biologically, the first process is we store energy long term. We use lipids to store energy longer than we use, say, in us, glycogen to store energy, or certainly longer than we use glucose to store energy. Fat layers are used as thermal protection. It's a great thermal protection. Cell membranes are composed of very, very modified lipids that are terrifically important. So we're going to find cell membranes produced from lipids. And then a bunch of hormones are made from modified lipids. All right. The names you'll hear: fats and oils, two different versions of the same idea, triglycerides; waxes, phospholipids, and steroids. I think waxes is fairly self-explaining. It's really just fats and oils that have been oxidized and joined to each other. So when they oxidize, they form wax. If you leave oil out a long time, it hardens and turns into a wax as it oxidizes in the atmosphere. Phospholipids are a key component of cell membranes, and steroids are hormones, steroid hormones. Right. So fats and oils are triglycerides. They consist of two components: glycerol, a 3-carbon alcohol, and then 3 fatty acids attached to them. When you make that, you created a thing called a triglyceride, triglycerol. So here's a picture. Here's a glycerol molecule. It's an alcohol. It's got hydroxyl groups on it, but no double bonded oxygen. If it had one of those, it'd be a sugar, but instead it's just an alcohol, hence glycerol. All right, and here's a fatty acid, carbon and hydrogen, and then it's got the acid bit at one end right here. A carbonyl plus a hydroxyl is a carboxyl group, so this is a carboxyl group. And we attach 3 of those fatty acids, and notice they don't have to be the same size, they can all be different, to the glycerol to make a triglyceride. So here's a triglyceride. This one is what's called a fully saturated triglyceride, and that means every carbon just has single bonds with its neighboring carbons, and it's saturated in the sense that it has as many hydrogens as is chemically possible. Absolutely saturated with hydrogens. There's no way to add more hydrogens on here unless we actually add another carbon, add another methyl group, and make the molecule bigger. So it is saturated. It's got all the hydrogen you can attach to it. Okay? These are the things that make up primarily fats. So butter, fat, and meats, and all of these is typically formed of saturated fatty acids that look like this. On the other hand, plants primarily use unsaturated fatty acids in their triglycerides, and what that means is that the fatty acid has at least one double bond somewhere in the chain, and so because that double bond, this carbon is bound to 1, 2, 3, 4 things, this carbon is bound to 1, 2, 3, 4 things, their valence chemistry is satisfied, but we don't have as many hydrogens as we could. It is unsaturated. And y'all hopefully know that all in all, unsaturated fatty acids are better for you than saturated fatty acids. These come from plants, and that's why there's such a push to reduce the amount of saturated fats that you take into your diet and increase the amount of unsaturated fats that you take into your diet, which come from plant sources. All right? And they can be monounsaturated, that just means there's a single double bond, or polyunsaturated, there would be 2 or 3 or more double bonds. It's usually just 2, sometimes 3. All right? An interesting feature is chemically these things stay liquid at room temperature. We call them oils, and they come from plants. These things typically solidify at room temperature. They melt if you get them hotter, but at room temperature, they're solid, so think butter, think fat, think lard. They're all solid at, normal room temperature because of this structure. All right. So the reason for the solidity or liquidity of it has to do with the shape of the fatty acids. Saturated fatty acids inevitably will be a straight molecule. Unsaturated fatty acids that are normally made by plants will have this kink in them like this. They'll have this kink where the double bond is right here, and it is a cis double bond. The rest of the molecules on the same side of the double bond, this cis. On the other hand, we have fatty acids that act like fats, called trans acids, there's no kink, and that's because they're trans bonds right here. The molecule comes off of opposite sides of the double bond, to the left and to the right here. Notice the difference. And so this ends up being a straight molecule right here. It solidifies at room temperature like fats do, and this is what we would call Crisco. Crisco is hydrogenated trans fat, all right? And you don't want to eat Crisco. It's actually worse than eating butter, from a health standpoint. This bond is really bad for you. The lack of double bonds is not bad in itself, but if you don't have enough of these in your diet, you run into trouble. You need these. You want these in your diet. What comes from olive oil, what comes from walnuts, what comes from fish, what comes from a number of dietary sources of fats that you have been told are good for you. Well, they all have those cis double bonds in them, and they're unsaturated, and you want those in your diet. You do not want these. These are metabolically actually quite bad for you. These are not bad for you, but too much starts getting into some difficulties with blood chemistry. So you got to keep the concentration of this stuff in your diet below a certain level to maintain good health. Avoid these. Don't eat trans fats. All right. So here's an example of an essential fatty acid. Notice the fatty acid chain is bent. Here's the acid end right here where it would attach to glycerol to make a triglyceride if we were doing. And it's got a double bond here, another double bond here, and another double bond here. In this particular case, this is an omega-3, omega-6, and omega-9 fatty acid all at the same time. The omega-3 and the omega-6, which just is count carbons, 1, 2, 3, 4, 5, 6, 7, 8, 9, count carbons, that's where the names come from. Omega-3 and omega-6 fatty acids are really important for you. Those are the ones you need for good long-term health. All right. Plants cover themselves in waxes. Well, they're masters at making these unsaturated fatty acids, and then if you oxidize those unsaturated fatty acids in the right way, you can get them to chain together along with alcohols to make this stuff called wax, and that's what creates the waterproof surface on plants. And of course, we use these waxes for all kinds of things. A lot of other organisms use them in certain ways as well. All right. Here is a really important molecule. So here's a phospholipid, and what we do with a phospholipid is we take this idea right here. Here's a triglyceride, and we take off, and this is what your body does. It takes off one of these fatty acids. So we have two fatty acids attached to glycerol. All right. And then in the third position, so here's your glycerol molecule. Here's one fatty acid attached to it. Here's the other fatty acid attached to it. On the third carbon of the glycerol, instead, we attach a phosphate group and then a short strand of amino acids right here. R is the rest of some amino acids right here. And all of this is going to be very, very charged. It's going to have negative and positive charges on the R group right here, negative on the phosphate, which is why it moves away. This is hydrophilic. The rest of this thing is hydrophobic. And so they isolate each other from each other right here, and you create this elongated molecule, and that is a phospholipid. And that's the key component of your cell membranes. All right. Here, this is a schematic version of it. This is a space-filling model of it, more accurate, what you would see under a microscope if you could really see the molecule. And here's a simplified version of it that we use in textbooks when we're talking about membranes, but not getting into the particular chemistry of the phospholipid. And the phospholipid typically has at least one kinked fatty acid. So this must have a double bond in it right there. And, it may have additional kinked fatty acids, or it may have unsaturated fatty acids, but it usually at least one unsaturated fatty acid in the phospholipid. All right. And so that makes it relatively fluid at room temperature, and membranes are very fluid. So here's a picture of a whole bunch of phospholipids. If you put them in water, they will self-organize like this. The fatty acid tails will all cluster together and face inwards towards the center of the cell membrane, and the heads will face the aqueous space inside the cell, typically drawn down in all of our pictures. So, if you had no other information right here, typically in this drawing right here, outside of the cell would be out here, inside of the cell would be down here. That's a convention when we make these drawings and show them on a TV screen or on a page in a book. All right? And this is a phospholipid bilayer. This is what all of your cell membranes are primarily made out of. And they automatically organize. If you take a whole bunch of these molecules, put them in salt water with a pH of about 7.3, and shake it up a whole bunch, after a while, all of the phospholipids will be organized just like this. Now, the last class of lipids that we need to think about are based on steroids. So this is still a hydrocarbon, but it's not a fatty acid anymore. You could call this bit, a hydrocarbon chain. It might have been a fatty acid once upon a time, but the acid group's been removed. But we have this hydrocarbon ring. So these are carbon-hydrogen rings. Notice, there's not much in the way of oxygen or phosphorus or nitrogen or sulfur showing up here. It's just carbon and hydrogen, so we can make rings of carbon and hydrogen. And this four-ring pattern, 1, 2, 3, 4, is real typical of steroid-based hormones. And the basic molecule is cholesterol. The stuff, you must have this. You've got to have this in your diet. It must exist in you, but everyone knows too much of this is a bad thing for us, all right? But we do use it in our diet, and we use it for two purposes. Cholesterol itself is used in cell membranes. They're tucked in here. Cholesterol molecules will be packed in these cell membranes in animals, therefore in our own cells, and it adds to the structural stability of the cell membrane, all right? And then the other thing that we do cholesterol is that we modify it. We've modified this chain right here, and replace this hydroxyl group with a carbonyl right here, and added a hydroxyl right here to turn cholesterol into cortisol, which is a hormone that is involved in fat metabolism and sugar metabolism in our biology. And we'll take cholesterol and we'll make testosterone in the same way. We'll take cholesterol and we'll make estrogen or progesterone. So, a number of steroid-based hormones are produced from cholesterol as the starting material. And again, we also use cholesterol as a key component in cell membranes by tucking cholesterol molecules all over the place in here. All right. Now let's take a look at proteins. Proteins are, as it says right here, one of the most abundant, organic molecules. The actual most abundant molecule on Earth is cellulose. And then next in line is going to be a couple of different proteins that are incredibly abundant, the ones used in photosynthetic activities primarily, that are just, by sheer ton, the most abundant on the planet after cellulose, which makes up the bulk of every tree. All right. Proteins, we do all kinds of stuff with them. We catalyze chemical reactions. That's probably the most, common process for amino acids. We also use them in sort of related, functions where they control chemical reactions and hormonal reactions in various ways. We use them as structural components in us. In animals, structural proteins are a big deal. That would be collagen and keratin particularly. There's others, but those are the two most abundant. Collagen is really fundamental to all living animals. Keratin is used more selectively depending on which group you're looking at. Same thing, protective structural keratin is primarily used as a protective protein on the outside. The dead, dry skin on the outside of your body is primarily made out of keratin. Transport the microtubules inside your cells, et cetera, et cetera. Any number of things. Poisons, a lot of them are proteins. All right. Those enzymatic proteins generally either break down things or put things together or cause chemical reactions to occur more quickly. Those are all versions of enzymatic functions that various proteins induce. We've got a whole bunch right here. You don't have to memorize these at this point. You do need to memorize the categories of function. So be able to list off these, that we've got proteins that function as enzymes. We've got proteins that function to transport stuff. We've got structural proteins. We've got hormone proteins. Your antibodies are all proteins. They're used in, immune defense. Skeletal muscle contraction, smooth muscle contraction, heart contraction are all based on contractile proteins. And we have a lot of proteins, including the albumin in your blood and albumin in egg white, that is used as a means of storing energy and amino acids. All right. So every amino acid consists of a central carbon and an acid group, a carboxyl group, double bonded oxygen, hydroxyl. When those are combined, when those are both attached to one carbon, the whole thing is called a carboxyl. And then here's the amine group right here. When we put this in water, this is an acid. This hydrogen goes into solution. When we put this in water, this is a base. It tends to grab a hydrogen, including that one, and attach it over here. And that's what makes an amino acid. And then there are 20 different biologically important amino acids, and they're all different by having a different R group right here. The thing on the side here might be just a hydrogen. It might be a hydroxyl group. It might be a sulfhydryl group. It might be an acid group. It might be a big carbon ring. It might be 2 big carbon rings. There's lots of choices, and there's 20 of them that are biologically important. And that's the R group, the side chain. And that's what makes one amino acid different from another amino acid. All right. We'll get into more detail on this later. But the amino acids come in some classes based on their chemistry. All right. So here's a table right here. So we've got the nonpolar aliphatic amino acids right here. All right, nonpolar means they don't dissolve in water. The amino acids themselves are all chained together, so the acidic nature is knocked out. The basic nature is knocked out because these things chain together to make a polymer. Here's the polymer. We neutralize an acid, we neutralize a base when we join them together to make this peptide bond right here. So the only unusual chemistry, once this is all done, is the double bonded oxygens cause this to still be fairly soluble in water. The nitrogen makes it still soluble in water. And it's the R groups that have varying chemistries. And that's what we're referring to when we look at this table right here. It's just the chemistry of the side groups. It has nothing to do with the the base or the acid of the amino acid itself. It's the side group. So all of these don't dissolve in water. Notice the lack of oxygens or phosphoruses or nitrogens. There's one sulfur right here, but it's in the middle, and this kind of makes it all neutralized. Here we've got polar ones right here. So there's oxygen, there's a sulfur at the end of a chain, very polar. There's a double bonded oxygen, very polar, and this nitrogen polar, etc., etc. All of these are pretty darn polar. There's a nitrogen right there, which makes this polar. Those are the polar side chains. Then we have ones where there's an amine right at the end, so that makes them basic in solution, right? We have ones that have an acid right at the end, so that makes them acidic in solution. So very soluble and basic, very soluble and acidic, just plain old soluble right here. And then what we've got is the aromatic, and that's where we have a ring on the end right here. Rings are aromatic hydrocarbons. These are going to be non-soluble, non-polar, and so they'll act a lot like this, but they take up more space. They fill bigger spaces right here, and so that kind of has an impact on the final shape of the protein. All right. So all of the amino acids have names. Valine and aspartic acid are two different amino acids. They've got single letter or 3-letter abbreviations that you see all over the place. So here we have their full names, but alanine is ALA, glycine is GLY, etc., etc., when you use them in their abbreviated forms. There are a few amino acids that you must get in your diet, and those are called the essential amino acids. So these are the ones you've got to get in your food. All right, there are 5 that we need when we're younger, at least 3 that we need when we're older. And what happens with these is we're made out of all 20 of these amino acids, but we can make the other amino acids in this list right here out of these. We can take these and turn them into the rest of these. So we can take isoleucine or leucine and convert it into all of these other guys right here with various complicated chemistries inside us. All right. So don't forget, just like all of the other monomers, we can join them together via dehydration synthesis. The specific bond is called a peptide bond, and it will include a double bonded oxygen hanging off from carbon to nitrogen here. All right. And similarly, we can have a hydrolytic reaction to break them apart. That's what happens in your digestive system. And you have a pepsidase that can break this bond right here by adding a hydrogen to the nitrogen and adding a hydroxyl group to this carbon, using up a water molecule in the process. Hydrolysis. All right. So we chain our amino acids together. We can make dipeptides and tripeptides, but most of them are going to be polypeptides. They're going to be really, really long. And when that polypeptide is folded up in the right form, the correct shape, then properly we call it a protein. But it's got to have the right shape. All right. So here's a illustration and animation of protein synthesis at work. And so protein synthesis is where we take amino acids and chain them together to make a polymer, a polypeptide, and then allow or guide or help that polypeptide fold up, as it naturally will, in water with a certain pH and a certain salinity, but guide it to ensure that it folds up into the right three-dimensional shape to take on a function as a functioning protein. So watch this animation. All right. Proteins, this is stuff students routinely get wrong and fail on tests, so please pay close attention to this bit. Every protein is a protein because it's taken on its proper shape, and the shape is the result of a series of sort of levels of organization. Right? The primary structure is simply the sequence of amino acids. That's what comes out of a ribosome as a protein is being synthesized. You make a polypeptide. The polypeptide chain is the primary structure. That's the information that's encoded in DNA, which amino acids are put together in what order. That's the information stored in DNA, transported by RNA, used by a ribosome, using RNA to generate a pattern, and expressing that pattern as a string of amino acids. And that's the primary structure of a protein. All right, there's the primary structure of two different protein strands right here. Notice the role of cysteine with its sulfhydryl groups hanging off in space as the R group. The hydrogens are stripped off, and we can bind the sulfurs directly to each other, satisfying their valence chemistry and making a sulfur bridge across here, either between two parts of the same polypeptide chain or between two different polypeptide chains right here to make a complex three-dimensional shape. All right. Okay. Protein shape determines function. So y'all are all familiar with the idea of hemoglobin being a protein that carries oxygen using a reactive iron atom right in the center of each hemoglobin molecule. But hemoglobin carries oxygen in our blood. Well, a change in the amino acid in one location from glutamic acid to valine, a different amino acid, due to a genetic mutation that's present, and causes a slightly different sequence of amino acids. So threonine, proline, valine, lysine, instead of threonine, proline, glutamic acid, lysine, and others. It's a much longer molecule, but this is just a short little 4 amino acid portion of the protein. Causes the protein to take on a very different shape, and that's the sickle cell version of hemoglobin, which unfortunately secondarily polymerizes to make a long wand inside the cell it's in, stretching the cell into a long, thin stick, which kind of bends into a sickle shape and clogs capillaries. So the function of hemoglobin has been altered by changing the sequence of amino acids. You've changed the shape of the protein, which changes its function. It still carries oxygen in us, but it also now creates this large structural thing, a sickle-shaped cell, which is not a good idea. We do not want that if we can avoid it. So here's some examples of those sickle-shaped cells. And, if you're curious what the problem is, these soft, squidgy, round cells tend to not get stuck on each other, and they slip through capillaries fairly easily. A capillary is a tiny circulatory vessel just the size of one cell, basically. These sickles can get jammed. A pile of them can get locked in there and block a capillary and therefore block blood flow to tissue. And if this happens enough, you have serious problems with anoxia, low oxygen in tissues, and tissue death as a result, along with all the pain and features that we feel, at the organismal level because of that. And that can lead to death in the most extreme cases. All right. The secondary structure of proteins is the result of taking that polypeptide and folding it in a particular way. And there are two secondary structures which are just repeating regular shapes that are included in most proteins that allow them to take on their final shape and allow them to do their functions. And those two are the alpha helix and the beta sheet. All right? So an alpha helix really is a helix. It just goes around and around and around again. The pattern of amino acids repeats over and over and over again, and naturally in salt water with a pH of about 7.3 and a saline concentration of about 300 milliosms, you'll get this nice predictable alpha helix. As this polypeptide is synthesized, it'll automatically curl up into this helix. In a beta-pleated sheet, a long strand has a complementary strand that base pairs to it, not base pairs to it, but bonds via hydrogen bonds to it, and then it folds again and again and again. So it's a longer, flatter variation on the same theme as this folded helix right here. And those give our proteins three-dimensional structure. Those are responsible for the conserved three-dimensional structure. And usually nothing interesting occurs biologically on an alpha helix or on a beta-pleated sheet. It is just a structure. It gives the three-dimensional protein some of its structure. All right? The tertiary structure is what happens in between alpha helixes and beta-pleated sheets. The bit that won't fold into a helix or won't fold into a sheet, that's usually where the action is. That's usually where the chemical reactions that this protein takes place in, if it is an enzymatic protein or an immune protein or a transport protein or all of these other things. It's usually that irregular area where something interesting is chemically happening, and that's the tertiary structure. All right? So, for instance, if the R groups, have a lot of charges on them that are all positives, they'll be repelled from each other, and they'll create a particular shape and behavior of the protein. Same things for hydrophobic, etc., etc., etc. All right? So here's some examples of tertiary structure. Notice there's no repetition. It's all unique, and that's kind of the whole point. All right? How do we get tertiary structure? Hydrogen bonds between side chains, sulfide bonds between side chains, hydrogen bonds. Here we've got a positive for a base and a negative for an acid, so ionic bonds right here. All of those bonds can set up this three-dimensional shape. And if you're, paying attention, you'll notice that the sulfide bond is a covalent bond, as is right here. That's a fairly strong bond. Next in strength is going to be these ionic bonds, moderately strong bond, and then finally these hydrogen bonds right here, and these weak hydrophobic interactions right here are the weakest of bonds. So this is the bit of the protein that can change shape most easily, and this bit right here most easily. Well, this bit is fairly rigid. Details of how proteins work. All right. And then finally, in the case of hemoglobin, for instance, we can get 4 different proteins and join them together. Each hemoglobin molecule is made out of 2 alpha proteins and 2 beta proteins. So 4 different proteins joined together, and that's the quaternary structure. That just means the 4th level of organization. Not all proteins have a quaternary structure. All proteins that do anything in your body have tertiary, secondary, and primary structures. All of them have that. All right, they all have primary, secondary, and tertiary structures. Whether they have quaternary structures just depends on the specific one you're talking about. Hemoglobin, yes. Collagen, yes. peptidase, an enzyme that, a protein that is an enzyme that digests proteins? No, there's no quaternary structure. It just depends on the particular protein. All right, primary structure, the chain. Secondary structure, sheets or helixes. Tertiary structure, unique three-dimensional folding patterns. Usually when we can apply the word protein, and this thing now has its function. It will do whatever it does. And then finally, if we take two or more proteins and join them together, we have quaternary level of structure. That varies depending on, it may or may not exist depending on the protein. All right. A little bit about proteins and shapes. So the shape is really important. If you change the shape, you change the structure and the behavior. An example of structure changing that we're familiar with is what's called denaturation of the proteins in albumin in eggs. When we heat the proteins up, tightly coiled globular proteins unwind into long filaments because they're really hot. All the hydrogen bonds making the globular protein have broken. So that's a whole bunch of secondary and tertiary bonds, in the structure. And then what happens is the individual albumin molecules bond to their neighbors, to their straight neighbors, and they make this big solid mesh. And they've taken on a fourth level of organization. There are a whole bunch of these things joined together, but not as blobs touching blobs, but as straight lines glued to other straight lines to make this white solid mass of cooked egg white. We haven't changed the amino acids. They're all still there. We haven't altered the primary structure. We've unraveled a lot of the secondary structure. We've certainly unraveled any tertiary structure, and we've created a new fourth level of structure when the strands are all glued to each other as the egg, cools. But that's, or as it heats first and then cools afterwards. But, a little bit about denaturation. So denaturation, to denature a protein is to unfold it. And if you unfold it enough, you can oftentimes not go back to its original state. That's what we're illustrating with this egg white here. It'll never work in its original form anymore. All right. The last class of macromolecules we want to talk about are nucleic acids. And the nucleic acids, of course, compose your genetic material. And then we also use them as energy transfer molecules in a whole bunch of different places. The genetic material, there's two types. Deoxyribose nucleic acid. Ribose is a 5-carbon sugar, but it's missing a hydroxyl group on it, hence the deoxyribose nucleic. Then there's ribose nucleic acid. So those are both the DNA and RNA, the two ways in which we store genetic information. All right, where do we find that stuff? The nucleus, mitochondria, chloroplasts, inside the nucleoid region of bacterial cells. All right. Roles of DNA, again, it's the genetic material. When you take all of the DNA collectively for a given cell, you refer to that as its genome. That's just all the information stored in the form of DNA. Chromatin is the name for DNA plus proteins surrounding it. That is what you really have in the nucleus of your cells. And a chromosome is that thread-like structure you can see when its DNA is packed up in anticipation of cell division. That's the one time we use the term chromosomes to refer to something we can actually see. DNA is primarily meant to store information. RNA is a means of taking that information with writing it down in a very similar alphabet called RNA instead of DNA. And RNA is used as a means of guiding protein synthesis. There's other jobs for RNA as well. Transfer RNA, ribosomal RNA, short interfering RNA. There's a whole bunch of different RNA molecules that do a whole lot of different jobs. The one you're normally thinking of when you think of RNA is what's called messenger RNA. That's the RNA that leaves the nucleus of a cell, attaches to a ribosome, and is used as the instructions for synthesizing a polypeptide chain, the raw material for a protein. These other guys right here are involved in that process. So a ribosome itself has some RNA in it. It's necessary for it to work. And the amino acids that are brought to a ribosome are brought by something called transfer RNA. So RNA is deeply, deeply involved in the process of making proteins. All right. when we look at nucleic acids, there are three pieces to each nucleic acid. There is a phosphate group, there is a ribose sugar, a pentose sugar. I said ribose. Ribose is an example of a pentose sugar. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5 carbons, and then there's an oxygen here at the apex. So this is ribose, or in this case deoxyribose, because it's got a hydrogen here. If it was ribose, it'd have an OH group right here. And then a nitrogenous base. And so the whole thing is a nucleic acid. The phosphate group, the sugar, and then the nitrogenous base. And in the nitrogenous base, we've got 4 different choices in DNA and 4 different choices in RNA. For DNA, it is C, T, A, and G. C, T, A, and G. Cysteine, thymine, adenine, and guanine are the 4 bases of DNA. If we're talking about RNA, then it's cysteine and uracil, which takes the place of thymine. Cysteine, uracil, adenine, and guanine still are used in RNA. So RNA is very much like DNA, except it has, RNA is just like DNA, except instead of having a deoxyribose sugar, here with the hydrogen, it's got a ribose sugar here with the hydroxyl group right here. That's one difference. And then instead of having, like DNA, having uracil, it will have thymine taking the role of uracil in RNA. So there are 2 chemical differences between RNA and DNA. Uracil instead of thymine, and ribose instead of deoxyribose. Okay? Those are the components of your nucleic acids. The nitrogenous base, there's choices. That's how we store information, by the sequence of these choices. The pentose sugar, which is always the same in DNA and always the other same thing in RNA, and then 1 or more phosphate groups. All right. We can also classify the nitrogenous bases if they're made out of 2 rings, guanine and adenine right here. They're called pyrimidines. That's the others right here. The single ring, they're called pyrimidines of a single ring. That's cysteine, thymine, and uracil. They have a single ring, the pyrimidines. The purines have 2 rings joined to each other, so that's adenine and guanine. And the way DNA works is that a purine will base pair to a pyrimidine. A pyrimidine will base pair to a purine. And hopefully you're already familiar with the rules. C's and G's go together, so cysteine and guanine go together. Adenine and thymine go together. A's and T's go together. Or in the case of RNA, adenine will bind to uracil. A and U go together, just like A and T go together in DNA. And those are the base pairing rules. One of these with only one of these with either of these, depending on whether it's DNA or RNA. And then that molecule self-assembles into this double helix right here, with adenine binding to thymine. This is DNA. Only DNA comes in the double helix. No RNA will look like this. And cysteine binding to guanine, etc., etc., etc. A last important idea, and this will be on the test, is the directionality of the molecules. It's a little hard to figure out at first, but once you get it, it's really easy. And the idea is that the 2 strands are anti-parallel to each other. So if I take my arm and I take my other arm, I can hold them parallel to each other like this, or I can arrange them anti-parallel to each other like this. And that's what's going on with DNA. The 2 DNA strands, they bind to each other in an anti-parallel way. And here's how we figure that out. Notice the sugar here. By convention, we can count the carbons, and this is carbon number 1, this is carbon number 2, this is carbon number 3. That's where this 3' comes from. 1, 2, 3. This is the 3' carbon and a hydroxyl group hanging off of it. 4, 5. 4, 5. This is the 5' carbon hanging off in space right here with the phosphate group attached to it, and that's the 5' end of the molecule. And so if one strand is 3 to 5 in this direction, the other strand must be 3 to 5 in the opposite direction. And that's the only thing you've got to know, is that they will always be in opposite directions to each other. Well, that's one thing you have to know. The next thing is the chemistry of DNA and RNA is such that you can only add on a new amino acid to the 3' end. So we can only build this molecule in this direction. We can add on going down this way. This molecule right here, the only way we can add on is to add on in that direction. And that creates some interesting complications in the process of DNA replication. The directionality of the molecule and the fact that to copy a chain of DNA, you've got to build some of it in one direction and you've got to build the other bit in the opposite direction, which introduces some problems. Now, notice the bonding right here. Remember, this is a rule. Learn it, memorize it. Adenine binds to thymine in DNA. That's the only two. They can't go. Adenine won't bind to guanine. Adenine won't normally bind to cytosine. They should only normally bind to thymine. And they are held together by 2 hydrogen bonds. They're held together with the strength of 2 hydrogen bonds. Cytosine and guanine normally only base pair to each other, and they're held together by 3 hydrogen bonds, so it's a 50% stronger bond. These guys are held together more tightly than A's and T's are held to each other, and that's an important aspect. That little difference right there is used in reading and expressing genes, in accessing the information stored in DNA. That little detail there turns out to be kind of important, so learn that. All right. So y'all all should know, that DNA can be rewritten as RNA. That process is called transcribing. Transcribe is to write something down in another format, but using the same language. You can hear English and then write English words down. That's an act of transcribing, like a court transcriptor. Court reporter is transcribing. Okay. And, the sequence is, going to be the opposite. So if we have a sequence written in DNA language right here, AATT, the transcription is UUAA, and then as we see, CGCG for GCGC. So that kind of covers all the bases for how, the two complement each other. And these two strands, as long as this is 5 to 3 in this direction, and this is 3 to 5 in this direction, can base pair to each other. They can stick to each other. Remember, they have to always be anti-parallel. DNA to DNA, anti-parallel. DNA to RNA, anti-parallel. RNA to RNA, anti-parallel. There's only one way in which they go together, and it's always anti-parallel. Always. Always. All right. We'll study this more in another unit, so this is not stuff you have to memorize, but just an illustration of the translation process. So on a ribosome, we use messenger RNA, which is, an RNA sequence, and reading sets of 3 RNA molecules is called a codon. That gives us the information for stringing amino acids together. And here's some transfer RNA, some ribosomal RNA, some messenger RNA, all involved in the process of making a polypeptide chain right here, which is a translation in a new language. We went from nucleic acids to amino acids. We translated the information in nucleic acids into a strand of amino acids, and that is translation. Transcription, translation. Okay. Where are we going to find our DNA? Nucleus, under normal circumstances, except when cells are duplicating. Where are we going to find RNA? Well, initially in the nucleus, because that's where you have to make it. You have to transcribe DNA in the nucleus, and then the RNA is moved out into the cytoplasm of the cell and is used, leaves the nucleus, and the cytoplasm is where translation will occur, taking RNA and making a polypeptide from that information. That process and that directionality of that process is referred to as the central dogma of information transfer in life. And the idea is that the information DNA is used to make RNA. The information in RNA is used to make proteins. We don't normally take proteins and alter RNA, and we don't take RNA and rewrite it backwards into DNA normally. There are a number of exceptions to this process, and that's a big, big deal in viruses. But for you and I, generally speaking, information travels only in one direction, from DNA to proteins. DNA changes over time due to mutation, which is errors in making the duplications, the replication of DNA, errors in making replications. When we have an error, like sickle cell, the sickle cell error, the translation process, the transcription process, the RNA chain is changed a little bit, and ultimately at translation we get a protein with a different amino acid in a particular location, and that's the sickling version of the hemoglobin protein. That information flows in that direction, and that is central dogma. Now, over the last 40 years, what we found is that there are a number of organisms, viruses, and certain genetic structures within us which can take RNA. There are a whole lot of RNA viruses that can rewrite their RNA as DNA and then have that DNA inserted into the nucleus of the cell. Those are called retroviruses. HIV is a retrovirus, that HIV being the virus that causes AIDS. Excuse me. COVID-19, the virus we're dealing right now, is an RNA virus, but it does not rewrite backwards as DNA, so it only exists as an RNA virus that codes for various proteins. But retroviruses start as RNA and get rewritten backwards as DNA, and that's a big, big deal. And that's the end of this recorded lesson on the macromolecules.